with this brief introduction, I invite Professor Yee to deliver a presentation on the medicinal mushrooms and her recent findings from her research, actually. Professor Yee, over to you. Stage is all yours. Thank you.
Thank you. Thank you. Thank you, Dr. Belshanoy. Thank you for having me. I'm pleased to be in among all of you. I think all of you are mushroom lovers, just like me and my friends in Malaysia. And it is always very exciting to speak to people who are like-minded, because sometimes I feel that some people do not understand why we are so passionate about mushroom. Mushroom is really not a plant, nor is it a vertebrate. It is a completely different family and kingdom on its own. And because it's so unique, so I think our interests are the same. And today I want to introduce you to something that I'm deeply passionate about. In Malaysia, we call it the tiger milk mushroom. I will explain to you shortly why we link this mushroom with tigers. It really has nothing much to do with tigers, but because we are also a nation where we are deeply rooted in tradition, and we have links with indigenous people who live in the forest. So we feel that we really need to uphold our tradition and the beliefs of our indigenous people, and hence the name tiger milk mushroom remains until today. But if you look at my title, Breathing New Life into Malaysia's Medicinal Gem, this is really something which I will explain why it is our Malaysian medicinal gem. And we have trademarked two names for it, not to be confused with other types of tiger milk mushroom. And I will explain this too. So we have trademarked xLr, which is the cold water extract, and trademark TM02, because this is really the strain that we have been working on for the past 15 years. And only this strain has been thoroughly scientifically investigated, not any other strains, for health and well-being. So I shall begin. This is tiger milk mushroom. In Malaysia, we are a peninsula, quite a small one. And in this country alone, in the peninsula, not even in our other side, in the east part of Malaysia, we find three very distinct tiger milk mushroom. The most studied species would be Lignosus rhinoceros. And within Lignosus rhinoceros, there are so many different strains. And hence, we have chosen one very fertile, highly medicinal strain, cultivated it, and did a lot of work on it. But in the midst of us discovering Lignosus rhinoceros in the peninsula, the mountainside of Malaysia, we also found actually there are two other strains, other species actually, and it is Lignosus tigris and Lignosus cameronensis. And if we do not look at the morphology of this mushroom, we would look at just, if you look at my mouse cursor, usually the indigenous people would pluck off the stem, remove the cap, and just sell us the sclerotium. Just by looking at the sclerotium, nobody would know that there are three distinct species. So what we did is that we look into the pores beneath the cap, and it is very different. So because of that, we also did genetic studies on it, and we found it to be distinctly different. So if someone comes to tell you that I'm taking tiger milk mushroom, the first question you should really ask is, what species? Because not all are the same. So a little bit of history. This is also history we discovered on our own. It is not something that is very well known. So back in 1664, this is documented in the National Archives, the diary of John Evelyn. You know, long before this part of the country was inhabited, we have traders coming through these straits, doing barter trades, businesses. And when traders come, they always want to take priceless valuables from the country that they visit. And one of such is something called Lac tigridis. It looks like a fungus. It is heavy. They were unsure if it is a metal or a rock. And all these were documented in the diary of John Evelyn. And they found the first taxonomical description. They named it Forms rhinocerotis, found here in Penang, up north in Malaysia. So as early as 1664, we already had history of this mushroom. And we also found out that the native people who live here near this cursor, my mouse cursor shows the mountainous area of Peninsular Malaysia, and the people here, the Semai people, the Temuan people, and of course other local people in Semenanjung communities. Semenanjung means the peninsula. They use it to treat asthma, cough, fever, cancer, liver-related illnesses, joint pains, and so on. Some use it for increase of vitality, especially after childbirth, and certain swellings. When I use the word swelling, I think it's rather loosely used, because for the indigenous people, they can't yet understand what swelling really means. But to us, it is a combination of inflammation, tumor, possibly low antioxidant. That's why it can proliferate so much. And we also found in Hong Kong, China, they also use it for quite similar activities to what we use it here in the peninsula. So Hong Kong, China is a little bit up north to this part of the map. Okay, so since 1964, when it was documented, nobody really knows what it is, and it has been forgotten for many hundreds of years until this year, 2002. I think our ex-Prime Minister, Tun Dr. Mahathir Mohamad, is quite famous around the world. So he was the first who actually brought this up again. He said he's a Western trained medical practitioner. So he's a doctor by profession. And in 2002, when he attended this convention for biotechnology, he mentioned that one of his friend gave him a rock-like structure for his chronic cough, because he had lung issues and chronic cough, and he couldn't get rid of it with any Western medication. So when his friend gave him this, it cured his chronic cough and lung infection. And it was in this convention that he challenged the scientists to investigate and analyze this effective mushroom. And lo and behold, would you believe it? Sometimes scientists do dabble with a bit of fate. Our father of tiger milk mushroom, you will see I will introduce his name later on, Dr. Tan Chung Sing. He was in this convention. And it was in this convention, after listening to Tun Dr. Mahathir, he was reminded that he had one of this rock-like structure at home. And his mother used to use it for him and his siblings whenever they were sick. So he went on a search to the forest to look for this thing again. And it looks something like this. So if you don't know that this is really a fungus, because it never appears to us as something mushroomy like. Unless you really go into the forest, you will see a structure like this. And this is buried underground, this sclerotium we call it. And this is really the part with medicinal value. And if you do a cross cut of this sclerotium, you will see that it is beige. If you grind it and add it to water, the color looks like very diluted milk. Although to me, I never taste any milk flavor in it, but some others tell me that it does taste like milk. So I think everybody's different. But the thing with this structure is that it can remain underground for months, years, or even decades. And it will only sprout when the environment around this mushroom is favorable, meaning to say that it is collecting all the nutrients, and when it senses that it itself is in danger, then it would sprout the stem and the cap, because below the cap is where you get the spores to propagate. And something unique about this mushroom is that you will only find one singular, and you'll find the next one 5 to 10 kilometers away. So this is why it is actually very, very rare. If we were to want to look for wild type, there are two things to consider. Number one, that it is very rare. Number two, as I've mentioned just now, what species are we taking? Not all tiger milk mushroom is actually the same, because we do have to identify the correct species for the correct bioactivity. And all this data has emerged since we have started working on it. So this is how it looks like. The wild type of tiger milk mushroom, it looks like this. But if I remember when we took this picture, this is actually very light. The whole structure is very light because all the nutrients that is found in the sclerotium has been used to sprout the stem and the cap. So by the time it is noticed, the stem and the cap, there is really not much nutrient found in the sclerotium anymore. You can still grind some powder out of it and try to use it, but really we upon our data generation from all our work, we noticed that the medicinal value is at least half gone. And because of that, it's very costly to buy full sclerotium, heavy, filled with components. But even when you buy that, now that we know it, you really have to get it alongside with the stem and cap. Otherwise, you really don't know what species you're actually consuming. So many years ago, before we started all this work, 15 years of work, we find that we have very few studies reported because the supply is just limited. But even if there was supply, between one sclerotium and another sclerotium, we can't find standardization of biocomponents. And that's what make this scientific studies very difficult, because all scientific studies require a standardized sample. So in 2009, 6 to 7 years down the road after 2002, Dr. Tan Chong Sing, our fellow in the Academy of Sciences, Malaysia, he managed to cultivate this. So you can see, he first grew the mycelium, he inoculated it, and grew, and this is the final product. So only this. You see, when he grew this sclerotium, because we understand how the mushroom works, how it grows, how it propagates, how it wants to replicate itself. So we do not allow the stem and the cap to form. The moment there is sclerotium grown, it is harvested. And this is grown in rice, not using the regular type of like sawdust. Some people grow it in wheat germs. None of those. We grow it in rice and other food-based materials. So everything from the start of cultivation to when we get the sclerotium, it is all food-based. And then as soon as it has matured, it takes about 6 months really for it to mature. Then we harvest. And we standardize this whole process so that we can get standardized component for downstream work. And this, I believe, for scientists like us, it is really important. We really cannot do an experiment when our sample biocomponents are fluctuating. So we really, really need a standardized component to start any studies. And this study has to be done using the same biocomponents throughout. And we have been using this standardized tiger milk mushroom. And that's the reason why we need to trademark and register this as TM02, because this is our most important reference when you want to talk about a real well-studied tiger milk mushroom. TM02 has been used for all our work since then. So this was my question earlier. Are you really taking the real tiger milk mushroom? Because I will also explain to you shortly that since the pandemic, in our country alone, there has been a boom of tiger milk mushroom consumption, because we found that it was really helpful for immunity and so on and so forth. I will explain this. So everybody says that they're taking tiger milk mushroom. And when I hear that, I always ask them, are you sure you are taking the correct species? And sometimes these things can get a bit awkward, but it's okay. I realize that as a scientist, we really want to consume the right thing, so it's okay for us to ask, right? So this is our data from our lab. We did actually compare it with the wild type. We don't take our validation very lightly. We have to ensure that our TM02 strain is similar to the wild type. It has real standardized biocomponents as the wild type. And it's just that TM02 is the best strain we can find to cultivate and do all the work. And we use ITS to validate the species. We also don't want to work with the wrong species and come up with various sets of data that gives us very conflicting results. So each time before we start the work, we actually revalidate the species again. So what did we as Medicinal Mushroom Research Group worked on? To be honest, really this name Medicinal Mushroom Research Group has only been created in the last decade. For the first 5 years of us working, we were just doing the regular scientist job, just wanted to know what this is, wanted to find out whether it was safe, and that is primary for us, because I came from a toxinology background. In my postgraduate studies, I did a lot on snake venom toxicity. So I am a toxinologist by training. So when someone asked me to come and look into tiger milk mushroom, to me, the first most important thing for me to consume anything that could potentially be good for my health would be it really needs to be safe. If it is not safe long term, how then can I say that it is good for my health? So that was a question that we had. So safety evaluation is primary. Before we even start to go and talk and search about what this mushroom can do, that is not our focus at that point. So we did safety evaluation for 3 to 4 years. It's not easy, because first and foremost, we had to find really committed people to help us with this. Because to do acute toxicity, it takes 7 days, which is fine. To do subacute, 28 days. We can hire somebody for 28 days straight and go into our animal housing unit and feed the animals and do this work. But what was really challenging to us, I remember, was the chronic toxicity. 180 days non-stop, including weekends, Saturday, Sunday. That's half a year almost. And we don't only have one cohort of animals, we have at least 5 cohorts because we need to touch on different dosages. So that is actually a very big, huge work on its own. And because we couldn't handle the harvest of these toxicity work, the harvest of the organs, the harvest of the blood, and all the other downstream tests, we needed to hire a few, 3, if I remember correctly, and they had to do it in different time frame. We had to stagger the work, and then we had to put in a lot of helpers to help them to harvest the organs, and then do pathological work, do biochemical work, uh, a lot of things we needed to do. We also, after doing chronic toxicity, we had to mate these animals to check on fertility and teratogenic. And then we had to put our samples through genotoxicity to show that it does not affect our genome. These days, we have a very interesting field of work called epigenetics. That means what we consume has the potential to modulate our gene expression. So we needed to ensure that tiger milk mushroom doesn't do that. So maybe if you allow me, I will stop using tiger milk mushroom. I will focus on using TM02. So TM02 does not do that. So after many, many months, years of toiling, we found that our anticipated dose that is really, really safe can be up to 10 grams per 60 kilogram of adult. And effective dosage, we really don't need to take 10 grams. There's a lot of mushroom powder to take. 0.5 gram, which is 500 milligram, is sufficient. Every day you take this 0.5 gram, it is enough. I've been taking it since I completed these studies. So if you do a very brief calculation, 10 years or more. So you can see that I did not start consuming TM02 the moment I saw the sclerotium. I had to show to myself that it is very safe. I had to look through all the data to tell myself that it is very safe. And then I dare to take. Now, everyone who knows me, my family, every one of them consumes TM02. And really, for a scientist, I think this is it's something that I proudly tell people. And that is it's really really good. But I haven't finished not even 25%, 75% of my story. There's a lot more things I need to tell you about this. I call it magical, because every single day when I hear feedback from people who consume TM02, it to me it's nothing short from magic. It's not to be taken lightly again. So as Dr. Bell has told you, because of our work for checking its safety, it was able to be registered with our Malaysian National Pharmaceutical Bureau in 2010 as a health supplement, a traditional health supplement with a registration number. All supplements and traditional medicinal in Malaysia has this MAL number, we call it the MAL number, to show to our community that it has already gone through very thorough checking and is marketed as free strike sclerotia powder. And because of this, we were awarded the ITEX and Geneva Award. So how then did we start looking into the bioactivities? So I would call this kind of work a reverse. It's very different how we did it. Usually, what the correct scientific process would be to discover the components, find out the bioactivity, put it through people to consume based on what we discovered, right? For TM02, we did it reverse because this is not something that is very new, so to speak. It has already been documented 400 to 500 years ago. And it has been known to be something very precious that people come to our nation to look for and took back to their own country. And because of that, we went to ask the native people or people who are already consuming, whether it's the wild type or not, why do they take TM024 or tiger milk mushroom? They told us this is a quality of life studies that we did in random. Actually, did it together with Ligno Biotech, which is my long-term industry collaborator. And this Ligno Biotech was really set up by the father of tiger milk mushroom, Dr. Tan, who cultivated it. So they told us that it relieved them from respiratory related illnesses, asthmatic symptoms, chronic cough, allergy, joint pains. It gives them more energy and again for growth, abnormal growth. So all this is just hearsay. I know there's a lot of products, natural products that is all based on hearsay or testimonial. So we call this QOL study somewhat a testimonial. And as scientists, we really want to generate data to show that these testimonials are real. So we went on to do extraction using various different type of methods. And we found one that is particularly useful and yielded very high bioactivity. And we named it the ACE technology, giving rise to the cold water extract of TM02. So we also named it xLr. X stands for extract. LR stands for Lignosus rhinoceros. So we have also trademarked this name and patented this technology. And so that brought about Essenti Nutrien and Nutrien Berhad. That is a startup company in the university. So this was TM02, the cultivated medicinal mushroom, safely assessed, verified by Ministry of Health. And we now have consistent supply. Then we put this material to extraction. We also patented this process of extraction using only water. So if you notice from when I explained the cultivation to you right through now, I've only mentioned food-based components, edible components. So this mushroom is grown in rice, which we eat every day. The components are all food-based. And now we extract it using pure water only, all devoid of chemicals. Not treated with high temperature, hence aqua cool, and all the active components are preserved. So we then freeze dry again this extract. It could be in liquid form or freeze dried form. And this is our whole process. So if it's TM02, it is the raw material. If it is xLr, it is the cold water extract components. So we also had the privilege to win several awards based on this technology, and hence Essenti Nutrien came about. So now that we have TM02, the raw material, which we did acute, subacute, chronic, and various human studies with it, we extracted it to do other bioassays and nutritional studies, chemical composition, and so on, which I will tell you about. So these two it goes hand in hand. I am taking both. Sometimes the TM02, I have taken the xLr, so they are from the same source. xLr is extracted from TM02. This is the raw material. This is the extract. Definitely with the extract, because it is more concentrated, you don't need as much grammage as you would the raw mushroom powder. So the first study we did was anti-inflammatory activity. So we investigated on the acute and chronic phases using our cold water extract, the xLr, and it showed that it possesses anti-acute inflammatory effect. So our extract was fractionated into different molecular weights. So we wanted to see which molecular weight was mainly contributing to the anti-inflammatory effect. So these are all our results. We find that the high molecular weight fraction, bigger molecules were doing the job, as well as the medium molecular fraction. It has a very strong inhibitory effect on TNF alpha production, which is a pro-inflammatory component. We also went on to use the extract to see the bronchodilatory effect, and we found that this bronchodilatory effect is mediated by calcium signaling pathway, both concentration and time dependent. And we believe that this would be one of the pathway that we will look at to see how it can relieve asthma and cough. And we look into the immunomodulating properties of TM02. We found that it was able, this is just a few of the cytokines that we thought was important, the IL-5, IL-6, MIP2, and TIMP1, because all these helps to improve our immune system and reduce inflammation, as well as to promote wound healing and reduce airway hyperactivity. So we thought this was important cytokines. You know how our immune system is really a very complex system. If I were to say that my immune system at this point is similar to the person next to me, that would be irrational. All of us have different body status, different inflammatory status, different cytokine levels, depending on our body, first and foremost, and the antigens that we are exposed to. But what is most important for TM02 is that it is able to immunomodulate. So whatever status we are at any one time, TM02 can help us to modulate and respond accordingly. So this is what we call adaptogens. It will adapt accordingly to each individual's microenvironment and help us to respond accordingly. So I don't know whether we are able to see this. Sometimes when I talk to people who are very into natural products, plants and so on, the question always come to why fungus? I think all of you here are fungal enthusiasts, because fungus, the genetic makeup of it is so close to human genetics, closer to than plant genetics. The similarity is higher percentage, and that's why when we use components from fungus and we consume them, it aids our body as if it is our own body that's producing these components. So I think this is a key important point for us to note. Why fungus will in the near future become what we call superfood. Or these are terms that we coin. Superfood, what does it mean? I also struggle to use this word superfood, because what kind of food is super, right? So, but it makes sense really why people come up with this, because if we consume something that is so similar to our genetic makeup, then it's as if we and this fungal species that we are taking are really hand in hand, synergistically working together to help our body to be healthier. I think that's what I'm trying to say. So important to note that interleukin 6 is the pivot in our immune system, because IL-6 can go both ways. It can then produce downstream pro-inflammatory cytokines, or it can go anti-proliferative cytokines that has anti-proliferative properties. So to me, anything that can touch IL-6 is very important to look at. So because of this, we were able to do, I call this a blessing, because, you know, because of our stringency of our pharmaceutical board in using anything natural, and then subsequently to allow it to go into medical centers to do clinical trial, is a task that is very big on its own. So for us, as scientists, to be given the ethics clearance to use a natural product such as TM02, to be given to patients with medical concerns, that in itself is already a victory for us as scientists. So we were given an ethics clearance to see whether TM02 can help for asthma. And we have completed these studies. It is in the midst of publication. So another thing interesting to note is that even though we have completed the studies, we know the results, we have the data, we are very convinced, but the world is yet to be convinced because there's not yet a publication. And to publish is yet also another hurdle for us. So, but it's okay. We try what we can, right? So what is important for us to note is that our data showed that all the patients who took TM02 reduce symptoms of coughing, wheezing, chest tightness, and dyspnea. And as usual, we also did an asthma control questionnaire, and every patient who were on TM02 told us that they feel so much better. And we didn't take what they say lightly. We can't just look at the external relief of symptoms, right? So we took their blood, we measured their exhaled nitric oxide measurements, and we did spirometry. So it all showed us that it has improved their lung function. We also did serum eosinophil studies, and it showed us that it has reduced, meaning to say that their level of allergic reaction towards anything, because asthma is a very, very inconclusive. Sometimes people who are asthmatic, they're allergic to smoke. Sometimes they're allergic to dust. Sometimes they're allergic to random things. They really cannot pinpoint what is the thing that causes them allergy. So we're able to reduce the serum eosinophil and also reduce the IgE. All this data really supports that it is able to control our reaction against allergen. Why so? You saw that in the previous slide. It is able to immunomodulate. And to me, as a scientist, and to the rest of the members of my research group, this is what we see. So the next one, we became a little bit braver after we were given clearance to do this clinical trial. We, because of the pandemic, and then people were looking for solutions to the problem, right? So we applied to do this work. Also, we are very grateful because Ligno Biotech Sendirian Berhad, our long-term industry collaborator, sponsored this work and given us a lot of help to be able to do this double-blind placebo trial on post-acute COVID-19 patients with respiratory conditions. So this trial is coming to an end in a few weeks' time. Okay, we have did some interim revelation to see what is the difference between the placebo treated and the TM02 treated. And again, we are very encouraged because those patients who had respiratory conditions due to the virus seem to respond better when they are on TM02. So we hope to be able to review this data soon. But you know, our bravery did not come just like that, just out of thin air, no. So we really had solid thoughts prior to saying that let's put this through patients. So our xLr components, again, we fractionated it into different molecular weights, the high, medium, and the low. And we did some in silico work. And then we did the ACE assay and the ACE2 assay. And we found that TM02 does really have the capacity to inhibit ACE2. Either it inhibits the active site or it completely stops the binding of the spike protein. So because this work has been published, you can refer to this publication. But this gave us a lot of background and confidence to be able to expect good outcomes in our clinical trial. And then we are going to move on to this. This study has not yet started, but I just put it here to show you that we are moving on with our clinical trials for allergic rhinitis. Because as I mentioned to you, again, allergic rhinitis is our body's inability to handle antigens that come. So we respond by having mucus in our nasal areas, coughing, and so on. So we strongly believe that this is yet another clinical trial that would give us good data. So we really, all our work really want to have only one single outcome. What is our outcome? We want to be able to tell people that we have something originated from our own country who is able to help very common problems that people face in the nation. Allergic rhinitis is one of them. Okay, so enough about anti-inflammatory and immunomodulating. The next activity that I will touch on is anti-cancer activity. This is not something very distinct from immunomodulation and anti-inflammatory because cancer has linkages with immunity and inflammation. But we wanted to understand if it is true or how could it be true that our indigenous people use it as help to swellings, tumor growth. So we investigated it on cell lines and we wanted to know which component in our xLr is responsible. So we find that majority of components that helps in reducing abnormal growth likely would be proteins. And majority of it is found in the medium molecular weight fraction. So this is more data to verify that what we see is true. These are in vitro data. We then linked it to in vivo data. We tested it on immune compromised rodents, and it showed that it would inhibit COX-2 activities downstream, and it could help to reduce tumor growth. In fact, there was a few rodents, I remember, that even when we gave them the cancer cell lines, we did a xenograft, right? The tumor just won't grow. So all my lab mates are very, very excited with this. And we also did a clinical trial on post-chemo breast cancer patients. So with cancer, we were not allowed to do it in conjunction with their treatment, simply because cancer is such a big, uh, I would say, a disease that is not yet well understood until today, because there are so many different biochemical pathway that is involved. And there's no one real source to anyone developing cancer. So I think it is fair for us not to be given the ethics to patients who are currently being treated. So we were only allowed to do it for post treatment. It's okay. We take it anyway because it's a chance for us to help cancer patients, because most post chemo cancer patients, even after treatment, they feel very tired. Their immunity is low. So we looked into that instead. And our results showed us that the mean fatigue score improved, systemic side effects improved. They were better emotionally and can function socially. And our oncologist also did a breast symptom score, which also improved. What I want to importantly stress based on this work is that these immuno-challenged patients who were treated with immunosuppressive drugs or anti-cancer drugs, as they would call it, their organs most of the time are somewhat compromised. Question always arises. If I give a cancer patient this natural product, will it deteriorate their organs further? And the answer is no. So if we look at the blood biochemical parameters, the liver function test, the renal function test, we can say now it is very safe to be consumed. Because this is the main worry. Because they have cancer, right? When they have cancer, they worry, if I take this, will it aggravate my cancer? Will it, I don't know, make my organs deteriorate? Then I can't function better. A lot of people like to say that if I take simply any natural products, it will make my body weaker. So we had wanted to show this. And we are very thankful, because we managed to get cancer patients who were open to take this and to join our clinical trial. That's again another blessing for our research group. So next, we want to move on to topical usage. So far, what we have talked about is biochemical systems that TM02 can help us with. We also believe that TM02 can work very, very well topically. So far, we have only been able to do in vitro assays. It can heal the wound. It can inhibit melanin formation, enhance collagen production. And how does it inhibit melanin formation? Is by giving rise to protection against UVB-induced toxicity. So we are still looking into this, but nonetheless, we have again obtained another clearance to make a cream out from xLr, which is the extract, and have started to try it on to people with eczema. Because again, I feel that or a topic this one atopic dermatitis. You see, people who suffer from asthma, most of them also have skin problems. This is again our body's immune response. So that is why we had wanted to do this project, which is still ongoing. But again, because of our excitement, we always want a before and after picture of certain uh, patients without revealing it fully. And we saw this. We saw improvements. Okay, so TM02 is really multifaceted, if you ask me. And to me, until today, after 15 years of working with it, I cannot put a finger onto a specific thing that TM02 can really help us with, because I feel that it can help us with a lot of health problems. The next thing that we will look into, which we are just touching on the surface, is using metabolic syndrome model. So I don't know whether our friends in India realize, but Malaysia is the fattest nation in Southeast Asia. Mainly because our food culture is very diverse and very heavy. I don't know whether I can use this word heavy. Our breakfasts are heavy, lunch is heavy, tea is heavy, tea both morning and afternoon tea. Yeah? And then we have dinner, which is heavy. And then we have, some people have supper, all heavy. And we eat, I think Malaysia is known as Southeast Asia's food haven. So all this gives rise to a lot of metabolic syndrome in our nation. And we want to see how TM02 can help. So one easier model that we can use is the type 2 diabetes mellitus rodent model, not the type 1. Because I know that there are some people who does this T2DM model and turning it into a T1DM by just failing the liver completely. And then they look at the effect. But in reality, if we were to apply this study to humans, it's not meant to be like this. Our liver slowly fails us, and pancreas and so on. It's a slow thing. Before we reach DM, we are suffering from MS, metabolic syndrome. So we are working on that, and we hope to be able to share some exciting results. So we so far we have talked about all the bioactivities, but before that we talk about the safety. And I want to say that we are not just doing things on the surface. We look into the genome, the transcriptome, the proteome of TM02. We really, really one day want to find out active pharmaceutical ingredients so that we can pick out some components, combine them synergistically, and see how it can work. So these are all very interesting proteins that we've discovered through our proteomic work. We also have our own genome database. We are the first group who derived the genome of TM02. We also did it for Lignosus tigris. We will soon do for Lignosus cameronensis. So we want to have the genome profile of all three tiger milk mushroom species in our nation to be able to see the difference. And we are looking at the protein. So as you saw just now, protein is one of the most important components. And we are looking for more. We recognize lectins, we recognize serotoplatinine and serine proteases, but we are looking for much more than that in search for new active pharmaceutical ingredients in the future. So with that, I think we have somewhat reached the end. I hope it has been enlightening for you as it is for me, and I'm happy to take any questions. Thank you.
Thank you, Professor Yee. I hope you can hear me.
Yes, I can.
That was an exciting presentation. Thank you very much for that. Now I invite our esteemed members in the audience to ask any questions or provide any comments if they have. Dr. Rohit Sharma, please.
Thank you, Dr. Shnoy, for the opportunity. Thank you, Dr. Yee. Very nice and informative talk. I think I'll take a couple of minutes to talk about your talk. So, very nice and extensive. Especially the toxicity experiments that you have told, very nice. They were very nice, and any person who wants to proceed can go ahead with that. Very nice. I have a couple of questions like, is there any difference between the content of the bioactive metabolite in the mycelial and in the fruiting body?
Okay, so we have had people who come to let us know that they are taking mycelium of tiger milk mushroom. It's just that traditionally, this is not the way they are consumed. I mean, we will never get wild type tiger milk mushroom in the mycelial form. That is something that is never been done before. So I know mycelium cultivation is because we have the technology, we put it in the bioreactor, right? But traditionally, this is not this has not been done. So that is why we didn't move through that route. So if you ask me, mycelium is likely structures that needs to still grow, be compacted to form sclerotium for it to have full bioactivity potential. So that's why we have never gone through that route.
Yeah, a couple of comments more. I think, as you said, post-pandemic, even I'll just share some of my personal experience that even my father is consuming here available product that is from the Ganoderma. So it is an immune kind of thing. So it is really good. And recently, one of my friend from Dubai just told me that his brother has been administered as a part of immunotherapy, the mushroom kind of thing for the cancer thing. So that what I have also from my personal experience, that what you suggested, that these are having a very good anti-cancer immunomodulative properties kind of thing. And lignocellulose, how it can be kind of different from the other like anti-cancer or kind of properties. Have you tried to compare with the other mushrooms also, the property of lignosus?
Yes. So in my laboratory, we don't just work with Lignosus family. We work with 3 Lignosus species as well as Cordyceps sinensis. We work with Chaga. We work with Sanghuang. We work with Entrodia. So we have done a comparison to see which is highly anti-proliferative for cancer cells. And we find that actually all of the 3 Lignosus species has that property. The next one would be Chaga, actually. So it's interesting, not all are good. And yet, if one is good for breast cancer, it may not be good for prostate and so on. So they are all different specificity. So I think it would take some time before we can narrow down to which particular medicinal mushroom is good for which type of cancer.
Thank you, Dr. Thank you so much for the questions. Thank you, Dr. Shinoy, for the opportunity.
Thank you, Dr. Rohit. Madam Professor E, can you please stop presenting, because then we'll get a better visual of you.
Sure.
Thank you. Yeah, great. Dr. Sunil Srivastava, please.
Yeah, Professor Lee, it was a very exciting talk. Thank you so much. I just want to know how easy or difficult is it to get to the fruiting body? Because this is naturally present in your forest. But if somebody wants to cultivate this, I mean, it's easy to grow a mushroom, but to get into this structure, which is actually dominant structure, so how easy it is to induce the sclerotial stage, and what you do in the lab, maybe if you're succeeding, you must be succeeding, then how, I mean, one can put it to commercial scale if somebody wants to do that. So how do you go about producing the sclerotial bodies? Because these are difficult bodies, I understand from my knowledge, because it's easy to grow mycelium, but if you have to induce the sclerotial stage to come about, and that too, I believe, as you said, this would be just a sprouting structure of the mushroom coming out of it. So it's a very, very specific stage, not a full dormant stage, because once the mushroom is also coming out, so it's exact stage which you pinpoint, and then you try to get the metabolite out. So how easy would it be if you were to grow commercially for this particular thing? Because which is very exciting. It has so many medicinal properties. I'm really very motivated by your talk. I mean, it's a wonder fungus, I must say.
Thank
What do
you.
you
Thank you
think?
for the question. Yeah. I would say it's not easy because
Yeah.
if it was easy, I think a lot of people would have done so prior to us doing it in 2002. So that is why this technique, we call it the solid state fermentation technique, has been patented as a company secret, trade
Mhm.
secret, I think, by Ligno Biotech Sendirian Berhad, which is the company that is founded by the father of tiger milk mushroom, Dr. Tan Chong Sing, our fellow in the Academy of Sciences. So we would like to really keep TM02 using the solid state fermentation technique as something that is unique for Malaysia, and for us to take the product of this cultivation method and share it with the world. That is the aim. So unfortunately, as a scientist, I don't even have the full data of how it is cultivated because it is already a trade secret. But I do know one thing though, it's really difficult to do the cultivation. So that is why I think even in the boom of using tiger milk mushroom in the pandemic, so people tried to sell the mycelium because they were unable to turn mycelium into sclerotium. There have been a few people who have approached us to say they have tried to grow halfway and then it didn't turn out. Yeah, so these are difficulties, and I believe it is also the same reason why upon the discovery in the ancient books, then it has been forgotten for many hundreds of years because it's not easy to do so. So these are things that I do acknowledge that is difficult, but that is why it became something so important for us to trademark and put protection on.
Yeah. But anyway, thank you for telling about this research. It's exciting, and I wish you all the very best. And
Thank you.
maybe it goes a long way to serve the humanity. Thank you.
Thank you so much.
Thank you, Dr. Srivastava. Dr. Chandra Mohan DS, please. Mr. Sachin Rajput, please.
Hello, thank you, ma'am. Thank you, sir. Ma'am, how do you figure out whether the phytochemicals or the active metabolite is produced by the fungi itself or the intracellular pathogen, which might be living in mutualistic relationship? How do you rule out that?
Mutualistic relationship with the environment, is that what you're saying?
I mean, the intracellular, whether it is living inside the mycelium or whether it is the fungi itself.
Okay. Correct me if I understand your questions correctly, whether the bioactive components are found in the mycelium or the sclerotium.
No. He wanted to know if there are some mutual partners of this fungus or this mushroom, which are endophytic, actually, kind of endophyte within the mushroom.
Yes, sir, exactly.
Do we have such kind of things in that particular mushroom? That's his
Uh,
question.
Dr. Bell, maybe you want to repeat the question to me once again?
Basically, he wants to know, are there any endophytes of this fungus? Whether some other forms of microbes living inside the mushroom, are they playing any role in the production of these metabolites? I mean, this is quite interesting. I'm not sure.
Okay, so secondary metabolites arising from the components of these mushrooms.
Basically, it's more like he wanted to know if any other microbial forms are having symbiotic relationship with this mushroom, and maybe he's talking about intracellular symbiosis, intercellular species. Yeah.
It's an interesting question. Okay, remember I mentioned to you that this mushroom grows singularly. It's a sole single one stalk of mushroom, never in schools. We believe that this is because if we look at the way the mushroom is found in the wild, one singular, another one 5 to 10 kilometers away, is because this mushroom only allow the fittest to grow. We have tried, although I didn't tell you in my talk, tried to see whether it was no, I can't use the word insecticidal, whether it has components that would kill microorganisms around it, or whether it can kill insecticides. We didn't find any. So but after cultivating this mushroom, we do microbial testing and so on and so forth. And we found not much. So maybe it doesn't have that property. We need to discover more on this. So 15 years for us is very short. I don't think I have answers to majority of the questions regarding this mushroom and its way of life.
Let me just ask my own question, if you permit me.
Yes, please do so.
The global market of all of these edible mushrooms or mushrooms is around $50 billion. So do you think we are going to recover the money we are going to invest in the medicinal research on the medicinal mushrooms? Because maybe you can make more money selling the edible mushrooms.
Yeah, it's true. I know. So how do I know this? Is because whenever I apply for grant to do research, they always ask me this question: what is the ROI? Nowadays, all research grant comes with the ROI. It's very difficult, I think, for scientists to research. Really takes a lot of effort and funding. And the basis of all fundamental research is time and money before we can reap economical benefits. So yeah, we still struggle with that. We hope to overcome that. I still don't know how to convince certain funders to give me the money to do the work that I want to do. But you're right.
No, the thing is, if you are doing taxonomic work, they ask us to do applied research. When we start doing applied research, we need lots of money. So it's quite of tricky, Anyway, thank you. Last question from Dr. Arundhati Sinha, please.
Hello, this is Dr. Arundhati Sinha. Very nice, awe-inspiring lecture, Dr. Professor. I have been abroad for some time working with trichoderma. I did very basic research, not so much high funder research. It was just a basic research, researching in my homeland, India, and as well as abroad in Ethiopia. So, ma'am, I was working on biological control on trichoderma. I am basically a mycologist, working since 2002. My PhD was in 2002. I wanted to ask, ma'am, regarding medical mushrooms and regarding the tiger milk mushroom, which you had just shown in your presentation. Where can I get more literature on it? See, mine is a very basic research, starting with the media, basic media, PDA and peptone dextroagar. At that time, 2002, way back, 22 years back, not much of genetics and all was available, isn't it? So that's why it's a very basic research of mine. I want to know, see, ma'am, by incorporating some very basic ingredients, like maybe rose Bengal, or maybe some other any natural things like kidney beans and all. Could we just grow this mushroom, tiger milk mushroom, or the recent research Lignosus, which you told, or the recent research medical mushrooms? That is my question, ma'am.
Yeah. So the first question is the literatures that if you want us to share with you, it is available. Some of them are open source, so you can easily obtain them. I think if you just Google tiger milk mushroom, you can find a lot. But please look out for, if you want a full extensive one specifically from our group, Then it has to be TM02 because that's our studied strain. And that is the one that has been commercialized and subsequently used in all our clinical trials. I actually can share with Dr. Bell later our list of publications on this, and then he can share it with the rest of us in the audience today. But using what type of media to grow tiger milk mushroom, I think that has been answered just now. We use rice and other food grain, food grade ingredients. So I think we don't use PDA. PDA is just in the beginning where we want to grow the mycelium prior to inoculation.
Yes, ma'am. Yes, ma'am. Thank you, ma'am. Thank you,
Thank
doctor.
you so much.
Any more questions? Maybe one last question from the audience. Just raise your hand. Dr. Chandramohan, please ask your question. Dr. Chandramohan, we can't hear you. Can you ask your question? Okay, Dr. Priyanka Shanay, last question from Dr. Priyanka Shanay. Dr. Pian Singh, please. Dr. Pian Singh, is there any technical issue? People are not speaking. Dr. Pian Singh, please. Dr. Priyanka, can you hear me? Dr. Roy?
So I can hear you.
Yes, please ask your question. Yeah.
Sir, it was really an amazing talk, madam. So I need to ask, is there any genome mining tools in order to find out secondary metabolites or bioactive compounds from this tiger mushrooms or any sort of other different variety of mushrooms? Similarly, in the case of an endophytes,
Yes. So when we
is
set
there any genome mining tools to? Yeah.
our genome mining tools are mainly freely accessible tools on the internet. We use all freewares because of the funding that we talked about just now. So we send our samples for long read sequencing because ours is we realize that we cannot do anything with short reads because we won't get sufficient data. So we use long reads, and subsequent to that, after getting the data, we will match it with anything that has similar. Otherwise, we have really, after trying that, we couldn't get much data. We did de novo sequencing. That's what we did. We did de novo sequencing for the DNA that we have isolated from this Lignosus family. If I may be honest, even though we have reported our de novo sequences, the first report was done in 2014, sometime around there. Until today, we are still mining from this data any interesting components, including secondary metabolites. And I don't think we can completely mine important metabolites until until my retirement, I believe, because as with big data, there's so much to do, and that is just genome. We haven't yet seen or fully mined data from the transcriptome. And then now with the advent of proteome, subsequently metabolome, and then other new bioinformatics techniques, we are consistently mining important data. And when we mine data, when we do in silico work, it also takes a long time and very powerful computing tools. So I think all these are in progress. We are looking forward to that. Fortunately for us, this new generation of students that I have, they are very computer literate because they are gaming students, right? They grew up playing games. So they love when I tell them, let's do something with the computer. And can you play with this software and that software? They're very happy to do it. Our new generation students who are now in their early 20s, they love doing computing. So I'm hopeful that we can get really nice data from our remining of our genome data and transcriptome and proteome.
Thank you, ma'am. Thank you so much.
Professor Yee, I'm very much tempted to ask allow the very last question, if you permit me.
Sure, sure. Please go ahead.
Dr. PN Singh, please ask your question.
Hello.
Yes,
Hello.
yes. Yeah.
Yeah, good morning, madam. Yeah, I'm PN Singh speaking here from Pune Agharkar Research Institute. So, like what you have showed in that is sclerotial, that medicinal use of this tiger mushroom. So, whether this aerial basidiocarp, that fruiting body of this aerial, this is also useful, like what you have studied in earlier from this sclerotial body of this tiger mushroom. I mean that aerial fruit body is having that tiger mushroom. Hello.
Okay. Yeah, yes, I can hear you. Maybe Dr. Bell can help to repeat the question for me.
Dr.
That
PN Singh.
aerial basidiocarp, I mean the fruiting body rather than this sclerotial fruit body, that aerial, that basidiocarp is having that tiger mushroom. So whether that basidiocarp is also useful, like what you have studied as
I think Dr. Rohit has already asked similar question at the beginning. Professor E, right? Mycelium versus sclerotia. He's asking about the fruiting body.
I mean that aerial fruit body is very nice fruit body having that, no?
Yes. So yes, people have asked me that same question. If I eat the stem and the cap, will it benefit me too? And again, we go back to tradition. And why do I always stress on tradition is because I strongly believe that our indigenous people, even though they have no science data to back their users up, I believe they really know a lot about this mushroom, much more than us as scientists. I always think that scientists is just, well, I can't say it's just because that would make us really lowly in grade. But I believe scientists and traditional healers, we need to go hand in hand. If you ask our traditional healers, why do they always just look at the sclerotium, never the cap or the stem? Because they really pluck it off and throw it away. It's because in the stem and the cap, there is not very many components that could give us medical benefits, only the sclerotium. So when we did this research, we would really want to give ROI to our funders and really focus on what is important and then move on to application. Once they see our application, I believe our fundamental studies can be funded much more. So we look through this route, look into the sclerotium, look into application, look into how we can grow the sclerotium and move on. While I can't deny that there may be uses for mycelium, for stem, for cap, but that's not our main focus at the moment because it doesn't return us a lot of applications since the traditional people don't focus on this too. Does that answer your question?
Anyway, yeah, I think you answered it very well, Professor E. Thank you very much. Yeah, thank you. I think if esteemed members in the audience have any more questions, please email it to Professor E. I have already shared her email address in the chat box. Please feel free to email her. She's a busy person. Maybe if she finds some time for you, she will reply to you. Professor E, thank you very much for spending for that wonderful presentation. More than that, thank you very much for spending your Sunday with us. I mean, we had some discussion about the dates. Saturday, Sunday, Monday. Yeah, you agreed for Sunday because most of us most of our friends get together, on Sundays or at 11 a.m. Indian Standard Time. Thank you very much for agreeing to that time and date. Thank you very much. It has been fantastic listening to your work, and we would like to have you with us for Micro Asia, Micro India, and AFB in future, events and future collaborations. Thank you very much. Yeah.
Thank you very much for having me. Thank you very much, everyone. Thank you.
Yeah, and I would like to thank Association of Fungal Biologists, Dr. Sunil Deshmukh, who couldn't make it today, maybe fully because of some personal issues. Dr. Shashireka, Dr. Mahavir Goswami, some wonderful people from the Association of Fungal Biologists for this collaboration. We are happy. We have had wonderful talks, exciting times collaborating with the Association of Fungal Biology. We got very interesting speakers who spoke about some interesting, for example, today's talk. For a taxonomist like me, it's an eye opener. It is a very different field, a different world there when you start trying to use or start getting into the application research or applied research. So it has been wonderful talking to you, Dr. Professor Yee. And thank you very much, ladies and gentlemen. With this, we sign off. And the audience has been wonderful as usual. Thank you very much for participating in today's talk. With this, we sign off. Thank you very much. Have a nice Sunday. Bye-bye, ladies and gentlemen.
Have a good day. Bye-bye.
Bye. Thank you, Professor.
Thank you. Bye.
Bye-bye.